Hi, I'm Marcus, and I'm doing my presentation on the influence of interspecific interactions on avoidance response to contamination. The study species for this were Danio zebrafish and Poecilia reticulata guppies. The study was done in Sao Paulo, Brazil at the Lorena School of Engineering. Just some quick background, fish can avoid contamination by moving spatially towards less contaminated habitats, and the contamination can affect the behavior of fish in terms of aggressiveness, competition, and spatial distribution. So their question was, do interspecific interactions between these fish change their patterns of avoidance to contamination? They wanted to assess if the avoidance responses of these fish populations, when exposed to a contamination gradient, were affected by interspecific interactions, and this was a controlled experiment. So the setup for this, they acclimated the fish to laboratory conditions for at least a week before performing any experiments on them. The contaminant that they used was copper. They mixed it with sulfuric acid and ultra pure water. And for the controls, they used sulfuric acid and ultra pure water just without the copper. And they created a non-forced chamber system out of borosilicate glass bottles, which they handcrafted for this experiment, which was pretty cool. And this is kind of what they looked like. So they were just like set up in a row, and they had seven different compartments for it. And there was two approaches that they used. There was a monospecific tests where they only had one species introduced to the system, and then a multi-specific test where both species were tested simultaneously. And they did this both for a control test and a contaminant test. So the control was just no copper, and the contaminant was with a copper gradient. All of the tests were done in a dark room. Observations were made every 30 minutes for 3 hours. These tests were performed in triplicate, so three tests for each one, three control monospecific, three contaminant monospecific, three multispecific controls, and then three contaminant multispecific tests. And the chambers were all connected, but they were sealed off with like plugs at the end of each test so that they could determine the final copper concentrations at the end of it. For when they were performing the experiment, there were the controls and the tests. For the controls, they filled the system just with plain water to ensure that the fish did not prefer any of the chambers. For the monospecific approach, they used 14 fish for each species, so two fish per chamber. And then for the multi-specific approach, this is when they put the species in together. They used seven of each species, so it was still 14 in total, but there was only one of each species per chamber. And then when they were doing the contaminant tests, um, they performed the monospecific tests with two different densities, 7 and 14 fish, just to see if their responses were affected by population densities. And their multi-specific tests were done with 14 fish, 7 of each species, like in the control. And their copper solutions that they prepared were created a concentration from 0 all the way up to 70 micrograms per liter, so one per chamber in the 7 chambers, and that created a gradient. So the methods that they used did seem relatively strong. They made sure that the conditions the fish were kept in were consistent throughout the entire experiment, which is great. The only thing is that they used different densities for just the monospecific test. They weren't really considering densities in their studies. That wasn't the point. So it isn't like a big deal or anything. They used the different densities during the monospecific test just to see if density had an effect on where the fishes went when contamination was present. And you'll see later on that they did get the expected result from that. So I understand why they only used different densities in the monospecific test. It was just to ensure that density wasn't a factor, like how they wanted to ensure in the controls that the fish weren't choosing some chambers over the others. But looking at the densities throughout would be like it would be a different experiment. I do think that it would have been useful to include these different densities throughout all of the tests just to keep it like consistent. Um, of course, that would like require more tests and more time and more fish and maybe more space. Um, if they were to like test more than just 7 and 14 fish, um, which would be a more expensive study. And ultimately, I doubt that including densities throughout all the tests would really change the results as much. But you never know. It could. Um, just maybe they could have included the different densities throughout all of the tests just to keep it consistent throughout. For their statistical analysis, they used a mixed design ANOVA to evaluate the distributions of the fish. They tested whether the variance of the repeated measurements, which was time, were consistent using Mauchly's test, and then used the greenhouse geyser correction when variability wasn't consistent. So basically, they were looking at differences across time. So every 30 minutes that they checked the chamber, they'd be looking at the differences between each 30 minutes. So 30 minutes to an hour, and then that 30 minutes to 1.5 hours in, and then like 1 hour to 1.5 hours, all the way through to the 3 hour mark. They were checking if the differences for all of the times were about the same using the Mauchly's test, because if they weren't the same, then that would violate the assumption of sphericity and the results would be skewed, which could give false positives. So in the cases where time was different than the rest of the other times, they would have to correct for that, and they did that using the greenhouse geyser correction. So that's some of the statistics that they used, and they also statistically significant differences for time or compartment pairwise comparisons were performed using Bonferroni post hoc tests, and if there was insignificant results, fish distribution data was pooled using the means for each of the times. So for the last bit of stats, they also wanted to know the number of avoiders and the avoidance percentage. They used number of expected organisms per chamber minus the number of observed organisms in each chamber. So when they were getting a number of expected organisms, they were taking however many were in that compartment plus however many were in the compartment adjacent to it with the higher concentration. So if like compartment number 4 had 3 fish, and then compartment number 5, which had a higher concentration, had 1 fish in it, then the total for compartment number 4 would be 4 fish. And then for the number of observed individuals, that was just the number of individuals in a given concentration and the individuals in all higher concentrations. So pretty much like the same thing, um, but it includes all of the chambers that are higher. So if you were looking at chamber 4, you would include the individuals in chamber 4, 5, 6, and 7 instead of just the one that's immediately adjacent. Um, And this is just because the individuals that were in the higher concentrations than the one that they were currently looking at were not considered avoiders, so they don't really count. And then in the control chamber, since there was no copper contamination, the number of avoiders was 0 because there was nothing to avoid. And then when they were doing the avoidance percentage, that's just the avoiders divided by the number of expected times 100. That's how you get that. And then at the bottom here is AC20, AC50, and AC80 values, and corresponding confidence intervals were calculated using PRI-Probit software. That's just the copper concentration that caused avoidance in 20%, 50%, and 80% of the exposed populations. For the results in their control, they found during the monospecific test that the distribution was random. So that confirmed that there were no external interferences or interactions. And in the control multispecific tests, they found that there was no variation across time, but for the guppies, there was variation across chambers where they tended to aggregate in some of the chambers together. Um, that just suggests that there was some sort of interspecific interaction response going on when they were put together, and the authors determined that this could possibly be explained by competition for space. For their contamination tests, they found that the densities did not influence their avoidance behaviors. In the monospecific tests, They both avoided the copper contamination, but it is important to note that guppies are more sensitive to copper than zebrafish, so they began to avoid the copper at lower concentrations. And in the multispecific tests, the avoidance of the zebrafish was less pronounced compared to the monospecific tests, so they were less likely to move away from the higher contamination concentrations when the guppies were present, suggesting that there are interspecific interactions at play. Here are some of the results. The top one is for the guppies, so you can see that the at the higher concentrations they had a higher avoidance percentage, and the same thing for the zebrafish, although it kind of is not as steep in all of it. And then this is with them together. You can see that the guppies are the black circles and the zebrafish are the open circles. The guppies avoided it more than the zebrafish. So just to summarize the results, in the multispecific scenarios where there were no contaminants, the species generally distributed themselves randomly across the chambers, with the guppies aggregating together. In the presence of contamination, the guppies were able to move to areas of lower concentration, whereas the zebrafish remained in areas of higher concentration despite having moved to areas of lower concentration when it was just them in the monospecific tests. The authors hypothesized that this change in avoidance response in zebrafish could have been caused by an increased aggression in the guppies, or it could have been less competitive ability of the zebrafish. It's also important to remember that guppies are more sensitive to copper, so it would make sense that they would want to be in the non-contaminated areas. If the contaminants did not affect their interspecific interactions, then we would have seen the same results that we saw in the monospecific tests. And this just demonstrates how the interspecific interactions are affected by the presence of contaminants. And for my personal assessment, I think that their results make sense. They align with what other studies have found on how pollutants affect fish behavior, fish dissection, and avoidance of contaminated areas and interactions between species. Besides just not having the methods be all the same with the different densities throughout all the replicates, I think that maybe they could have used a larger sample size just because 7 and 14 fish aren't that many. But I think that they got meaningful results either way. It would be interesting to test this with other species and with other contaminants just to see how like their results would change across different species and contaminants. The authors did touch on a lot of the possible alternative hypotheses. They grouped them into two groups, which is either the effect of guppies on displacement of zebrafish or the effect of contaminants on the displacement of zebrafish. So it was kind of difficult to come up with an alternative hypothesis that did not fall into one of those categories. I thought maybe it could have been based on the strength of reaction to copper, so the guppies could have just reacted sooner and stronger than the zebrafish and took up all of the space in the lower contaminated areas before the zebrafish could respond. It also could have just been a behavior response that was not aggression or lack of competitiveness, but any other sort of like behavioral response would end up falling into their the two categories of hypotheses of either the guppies being the reason the zebrafish were displaced or the contaminants being the reason the zebrafish were displaced. So yeah, I think that this was overall a pretty strong study. It was well done, it was thorough, and they seem to have covered all of the bases with that. So yeah, um, thank you. That is my presentation.